Hello, everyone. Excited to be here to present for the first time at WBDS Latin America. Today, I will be talking to you about a new web application that we developed, MolEvolvR, to characterize proteins using molecular evolution and phylogeny. This app was built by three fantastic undergraduates, Sam, Lauren, and Joe, in my group. We use molecular evolution and phylogeny to characterize proteins and protein families, especially in understudied pathogenic groups, because we can learn a lot of things about the biology and function of these protein families by taking such a careful evolutionary approach. We have created a streamlined approach for that is widely applicable across proteins and genomes of interest. We are testing our web app and our companion R package that we're also developing on critical pathogens from Bacillus anthracis, Mycobacteria staph, and Vibrio cholerae. So let me take you through what we have done recently. So this is a workflow that underlies MolEvolvR. People can start their analysis with one or more protein sequences of interest, and they can find a comprehensive set of homologs across the tree of life, domain architectures, including secondary structure predictions and cellular localizations of these different proteins for every one of their homologs. And finally, you can also do a carefully crafted phylogenetic analysis for each and every one of these proteins. If you have done your a little bit of your analysis in other tools such as BLAST or InterProScan, you can also bring in those output files for further characterization within our app. So here on the right, I've shown a few data upload options. You can start with FASTA sequences or accession numbers, MSA, or any of the other outputs that I mentioned. We look at not just whole sequence analysis, but also we find homologs by domains, and we give you the option to customize your analysis. For the domain architecture analysis, we look at several different databases and for profiles, as well as prediction algorithms for transmembrane region, cellular localization, and signal peptides. So here in the top left, I've shown a cartoon that shows representative homologs that have different domain architectures in Gene3D and PFAM. We also look at the phyletic spread of not just the proteins, but also the underlying domain architectures across different lineages, and that is shown in the heat map here. We use an upset plot, which is basically looking at multiple intersecting sets. And here we are looking at how different domains are coming together across different proteins in the different homologs. So here, for instance, the tall red bar is showing us that the combination of SIG and TOS track, that domain fusion is what is appearing most frequently amongst all the homologs. And finally, we have also found that when we are looking at groups of proteins or co-transcribed genes in an operon, it often helps to look at not just individual proteins, but all of them in a context together. So we construct a domain proximity network here, and we connect domains if they have co-occurred within the same protein, and the size corresponds to how many times this occurs across each of the homologs. And finally, we also do the phylogenetic analysis by constructing multiple sequence alignment for each and every one of the proteins, as well as a color-coded phylogenetic tree by lineage. We also look at the phyletic spreads and look at which are the lineages in which each of these proteins and their domains are present. In summary, we have developed a generalized computational evolutionary approach to study protein families, and we have characterized them in terms of their homologs and their domain architectures. We have tested this on several operons across bacterial pathogens from Staph, Vibrio, and Bacillus, and few of these are already published as preprints or in frontiers. We also have a fully working prototype that you should definitely check out. It's in the PSP Evolution web app, and we also have a companion preprint, and that's currently under review now. And finally, please do not forget to check out our main web application that I've presented today, MolEvolvR. You can look for it in JRavi Lab. Oops, there is a typo here. It's jravilab.org/MolEvolvR. We are also developing a companion R package so that we allow for customization for computational biologists. I would like to thank my funding sources. And once again, I would like to thank WBDS for this fantastic opportunity. Please reach out to me if you have any questions about how to use the app, if you want to incorporate any other tools of yours within our web app, or if you want to start a collaboration. We look forward to hearing from you. And thank you for stopping by our poster. Have a great day and enjoy the conference.